Evolution describes all of the changes that have transformed life on Earth, starting from the most basic early beginnings to the wide diversity of life that we see today. Overall, there's two major categories: microevolution, which is a small scale affecting a single population, and macroevolution, which is a large scale affecting change in species across populations. Today we're going to focus on microevolution. Now, evolution comes from a combination of genetics and environmental factors. Genetics create variation, create differences, and some differences are better suited to environments than others, and those that are better suited survive and reproduce, and hence evolve to meet the needs of the ecosystem they live in. Genetics really underlie the continuity of life on Earth, and we know that all life that has ever existed on Earth shares at least two characteristics. They have nucleic acids, be it DNA or RNA, and they have proteins with a universal genetic code to do the work of the cell. If you were to take a gene from a bacteria and put it into a human, you would still be able to make a protein. Not necessarily functioning, but it would be able to be read. This is true because we all share a LUCA, a last universal common ancestor, the most recent organism which every form of life shares a common descent from. We don't really know very much about the LUCA. We know that it had to have been single-celled and prokaryotic because it was way before any of those changes occurred. We also know that it had to have been heterotrophic. It ate other cells, or LUCAs probably, and we knew that it had to be asexually reproductive because it's way before sex evolved. Now, could there have been more than one organism alive at the time? Yeah, but the LUCA is the last one that every life form on the planet has a connection to. There are many sources of genetic variability, and variation is the key to evolution, because if there's not variation, species cannot be selected for, species cannot evolve. Natural selection is probably the most obvious, where you have some sort of selective pressure based off of some sort of trait, and the most favorable phenotype will survive and reproduce. Artificial selection is a human-caused version of this. It's much faster than natural selection because there's no randomness to it. There's almost all of our domesticated animals and most crop plants are the result of it. Hybridization, selective breeding, some sort of artificial selection. You can see some examples of this on the right. Mutations are a major source of variation, but it's a random change. It's not selective, it's not adaptive, but it creates a lot of variation. Genetic drift is another source of genetic variability. It's a random change in frequencies due to chance events. The founder effect, a new population is established by a very small number of individuals whose genes dominate the gene pool. Example of this is on the top right. The bottleneck effect, some sort of catastrophic event, is going to result in a sharp reduction in the size of population due to environmental effects, human activities, something like that. And the population is no longer representative of what it once was. You can see this example in the bottle metaphor on the bottom. Gene flow is migration of individuals. It's an equalizing change. I don't really have an example of that here, but it balances traits out. If you look at, say, human skin color, as we start moving between areas, skin color is favoring a medium. Sexual selection is the last major source of variation that we're going to talk about, and basically it's non-random mating. Males do displays to attract different females, and that's going to affect mate choice. You have intersexual and intrasexual competition where people fight over mates. Sexual selection is definitely a major source of variation, but not necessarily always a positive one. Sometimes traits that are selected for in mating are not very good for survival. Once selection is actually beginning to occur, there's really three patterns. If you start off with a normal bell curve of any polygenic trait, you could have directional selection, which is going to shift towards one extreme, say favoring the black moths at the bottom. You have stabilizing selection, where you're going to favor the mean or the intermediate or the gray moths in this example, or disruptive selection, where you're going to go towards either extreme. So instead of going for the gray, I'm going to go for the black and the white moths. Over time, biologists have really tried to unify the ideas of Darwin and Mendel, and we call this idea NeoDarwinism. We know that evolution is a change in the allele frequency within the gene pool of a population over time. At least that's the current definition of evolution. So it's way beyond what Darwin would have known and incorporates a lot of his ideas into the ideas of genetics. But some of the words that we use are a little bit different. We need to understand that a population in NeoDarwinism is a localized group that is breeding within itself. A gene pool is the collection of all the alleles in a population, all the genes of all of the individuals in a population, whereas allele frequency is how common is that allele in the population, how many dominant A alleles versus how many recessive A alleles in that whole population. You need some understanding of these words within an evolution perspective going forward. Now that we have some understanding of the terms going on here, we can look at how we actually measure evolution. Hardy and Weinberg, the two guys shown here on the right, independently came up with a theory in 1908, a formula, a mathematical basis for population genetics. Basically, it serves as a null hypothesis. The null hypothesis is the hypothesis that there is no relationship, there is no change. It's very hard to show if a population is evolving, but if we can mathematically show that it is not evolving, that gives us some really good information. There are two major parts to this formula. You can see them on the right. The P represents the dominant allele, whereas the Q represents the recessive allele. So P plus Q is equal to 1 is my gene pool population. And if you look at P squared, well, I've got 2 of the P's, therefore that's my homozygous dominant. 2PQ is my heterozygous, and Q squared is my homozygous recessive, adding up to 100% of the population. Now, Hardy-Weinberg equilibrium has 5 conditions, and if you look at the sources of variation, they make sense. Now, the first one is that there must be a large population so that you don't have genetic drift, no random effects causing change, no migration, no gene flow, no balancing of the traits. Random mating means I'm going to have no sexual selection, no natural or artificial selection, and no mutations in the gene pool. If you take out all of those sources of variation, there will be no evolution. In Darwin's book on the origin of species, he didn't actually talk about, well, how species originate. It's a lot harder idea than it sounds. There are currently more than 20 different definitions for what is a species. We're going to mainly focus on the biology one because this is biology, and that says basically that if the organisms can interbreed to produce viable and fertile offspring, it's a species. Viable means that they survive, fertile that they can breed. This is true because that means they share a common gene pool, which does mean that the biological species concept is based only on gene flow. Because of that shared gene pool, a genetic change that occurs in one individual can spread through the population as that individual and its offspring mate with other individuals. Yeah, but there's some problems with that. What about asexual ones? What about preserved specimens? I can't breed those. Fossils. This isn't Pokemon. I can't breed fossils. So we have a lot of other definitions. We're going to look at a couple of them. Morphological species concept, basically the body shape, size, and physical structures. The big problem with that is analogous structures. Ecological species concept, a species is defined by its niche. So are all parasites the same species? Probably not. So it's useful when you're studying ecology things, but not really when you're looking at biology. The phylogenetic species concept is based off of common ancestry and monophyletic trees, which we'll look at later. And all of these different definitions have their uses. If there was one definition that answered all of the problems, there would only be one definition, and we have more than 20. The most basic idea to speciation, though, is reproductive isolation according to biological species concept. If you look at it, there's really 2 major ways that this can happen. There are other situational ones, but allopatric speciation or different country speciation, some sort of geographical barrier is going to come up. A cavern, a road is built, a field is cleared, something. And the species become reproductively isolated because they are separate and they're no longer interbreeding. And given enough time, they'll become different species. Sympatric speciation is a lot more complicated. There must be some other form of isolating mechanism that occurs. So in sympatric speciation, there are a lot of different isolating mechanisms. We're going to look at the major ones here. They fall into 2 major categories, the prezygotic or prior to fertilization and the postzygotic after fertilization. Habitat isolation, they live in different areas, say land versus water. Temporal isolation, they mate at different times, winter versus spring, day versus night. Behavioral isolation, they have unique behavioral patterns and mating rituals, say the blue-footed booby and their dance to attract females. Mechanical isolation, they have different reproductive anatomy, so say changes in the curvature of snail shells. Gametic isolation, the proteins on the surface do not allow fusion. So say eggs and sperm in the ocean, they're not going to connect, they're not going to fuse, they're not going to fertilize unless they are the same species, so that it works. After you've had a mating attempt and you had postzygotic barriers coming in, basically you've made an organism, there's really only 3 major isolating mechanisms left. If you remember, the biological species concept needs you to produce viable and fertile offspring. So reduced hybrid viability, that hybrid is weak and it doesn't survive. Reduced hybrid fertility, the hybrid is sterile, say a mule. Or hybrid breakdown, this one's probably the hardest to see, but if you look at the wheat on the right, the big bunch is the original one, and then the second generation is on the left, and the third generation is in the middle. Those strong hybrid traits can go away over time.